Hello, everyone. Today we're going to be talking about how to write a good results and discussion section. So while we're specifically going to be looking at writing for organic chemistry, it is worth noting that a lot of what I'm going to be presenting today applies to any sort of scientific writing. So when we're looking at a results and discussion section, it's worth noting that these two sections have slightly different goals, but they're often quite complementary. So whenever I'm looking at presenting my results, what I want to do is clearly describe what I found. And we want to do this in a nice, concise manner because we don't want to ramble on too much, but we need to summarize every key point of evidence that I have. So whenever you're writing this, one of the big things you should focus on is what did I find? So anything that you found, you should go ahead and summarize here. And everything that you write should answer this question. It is worth noting that the answer to these sections often include figures. I found evidence in this figure. And we'll look at that a little bit more detail in the next mini presentation that I have of how we can merge our writing and our figures. But once I've gone ahead and presented, okay, here's what I found, I need to also put a little bit of an interpretation on it. So what do these findings mean? What's their significance? Why should I care? What do they tell me? And it is worth noting that oftentimes we want to keep these two things separate. We want to be very clear what's fact and what's opinion. But sometimes, in order to get a good narrative, we often want to bring these two together. And because, again, one of the big things to remember when you're writing a paper is you're trying to generate a narrative, a story. So whenever you're writing the results, you should always be thinking about the discussion. And whenever you're writing the discussion, you should always be thinking about your results, no matter how these two are connected. Now, let's go ahead and look a little bit more on how we connect these two things. So let's say I made three major measurements. So I got three major results to present. So there's going to be two major ways to present these findings. So the first one is what's called a traditional blocked format. I'm going to present all three of my results, one right after another, and this will form the traditional results section. So this takes the form of results 1, results 2, results 3. So once I've summarized all of my results, I can go ahead and discuss each one individually. So you'll discuss your finding from your first finding, second finding, third finding, and so forth, each one sequentially. And again, do make sure you present your results in the same order you generated your discussion. The other format is what I like to call the iterative format. So I present each result and then I discuss it. Second result, discuss that. Third result, discuss that. And this way they blend together a little bit more smoothly, but it can be a little more difficult to figure out what's results and what's discussion. And you can often identify when somebody's used the iterative format, because this section will be titled results and discussion, as opposed to results and then a discussion section. So whenever you're going to be presenting your results, you want to make sure to split this part into several individual portions. So the first one is whenever you're presenting your results, you should always set the stage. So I don't want to just suddenly hit the people with data. I need to let them know what the data is. So remind the reader how you obtained your results. So for example, if I'm looking at TLC data, I need to let students know I collected a TLC using the solvent and these standards. That way they know I've set the stage so they know exactly what they're looking at. Then you're going to want to refer the reader to any graphics. So oftentimes this is going to, for a lot of what we're working with, this is going to take the form of tables. But sometimes this could be a figure, such as a picture of a TLC plate, or presenting, as in some of our later sections, any sort of spectrographic result. So all of these are going to be good graphics that are often worth 1,000 words. So you need to go ahead and let the reader know exactly what graphics you're looking at and where we can see these results summarized. So then you're going to need to fill in the details of what we found. So identify any key findings. So for example, if you're looking at a TLC, you're going to want to summarize something like, what were the RF factors? Did I have a narrow peak, a broad peak? Again, pointing the reader's eye to the important things. Make sure to describe any important trends. So for example, if I have three peaks in three spots in a TLC, point which one was furthest, which one's middle, which one didn't go, which one was the lowest. This way it will help the reader to interpret, okay, here's what the intermolecular forces of the individual species are. You can start to see how the evidence comes in. Now, here's one of the last important points of results. Make sure you include any sort of unexpected results. Sometimes this is a good unexpected result, like we had a rather large yield. This could be a very bad unexpected result of, turns out my melting point had a very wide range. I have a very impure species. So again, pointing out general trends, and it is worth noting that this can often get into a little bit more discussion. So again, just kind of point out high, low, bring the reader's eye to the important points. Now, one of the important things is that this takes place for each and every result you make. And it is worth noting, sometimes you can combine these individual points into single sentences, but sometimes they need a little bit more room to breathe and sink in. But again, if you've got 3 results, you need to go through this whole process at least 3 separate times. So keep that in mind, how many sections you need. Now, how do we go ahead and work in the discussion section? So it is worth noting there are 2 major parts of the discussions. So the first is going to be discussing the results. So this is going to be discussing the specific results in your section. So knowing what discussion pairs with which results. This is most particularly problematic if you're using a block format. So if you're using a block format, your discussion is separate from your results. So you should start with quick reminder of what your results were. So when examining our TLC data, we found, and again, short sentence summary. That way the reader doesn't have to go back to the previous section. So this can be followed by an interpretation of the results. So specifically looking at potential explanations for the results and telling the reader a lot more about the significance. Now, one of the important things is it's often important that if you're doing any sort of connection to a larger context, you should include references to the literature. So for example, when we're looking at caffeine content in tea, you might want to make a reference to how much caffeine content should have been in the amount of tea you had. And for that, you're going to need some sort of reference to support that data. And again, remember, any point you bring up in your results or discussion should be supported by data, either numerical, observations, or literature. And you're going to need to go through these key steps for each and every one of your results. Now, once you've summarized and discussed each of your results independently, you need to go ahead and kind of bring them together. So this is often a concluding remark. So you need to summarize the important points. And what's the overall implication? Did things go well? Did they go poorly? This is going to be very big picture. Now, some papers, this is going to be given its own section of conclusions and summary, a conclusion section or a summary section. And sometimes the conclusions or summary is just the single paragraph summary of your summary. So again, the very important points if somebody went right to the very end. Now, when we're talking about this overall discussion, some of the big points you're going to want to consider is, first of all, what was the overall success of my work? Did I have high yields, low yields, good purity, poor purity? This is going to be one of the first questions you should answer. And again, always support with evidence. What were the implications of this work? Does this have any broader impact? Is there any big take-home points? Did I gain any new insights? Did I discover something about how easy or hard it is to separate caffeine? And did you gain any additional understanding? And again, always an important thing, practical applications of the work. So when you're doing a separation, is there things that you should take into account? Is there things that would be important if you were going to scale this up? So, and again, less important for this specific work, but how will this work impact society? So especially when you start working your way to real scientific publications, this is always something you want to keep in mind. And one of the big things to remember is when you're finishing any discussion section, you always want to zoom out, take a bigger look at everything you did, and put it all in context. So again, results, you're looking at everything one at a time, summarizing important findings, and then discussing each of those points individually. Then zoom out, bring them all together, broader implications, broader evaluations. All right, hopefully this was helpful, and see you in lab.